Welcome to the University of Illinois College of Medicine in Peoria. I'm Shannon Egley. I'm anatomy faculty here at the university. I'm also the anatomical coordinator at Jump Simulation here in Peoria. And welcome to Microscopy 103. I'm assuming you've watched 101, where you got a chance to put your scope together, get to know some of your lab equipment, maybe get a toolbox full of tools. Then 102, where you perfect some skills. Nah, I shouldn't say perfect. You practice some skills and learn some skills, things like understanding contrast, resolution, magnification. So you understand what the microscope can do, and now the third part, and from here on out, is taking a topic and using your skills to learn more about the topic or to see the invisible world that you couldn't see before. So today we're going to talk about the anatomy of cells, and I figured where's a better place than to come to the microscopy lab at the university, where medical students are learning to look at cells that are disease-causing cells, so pathogens, looking at what happens as a result of that, so pathology, so like cancer cells or diseases that happen to human cells, and then also look at some healthy cells like muscle cells, heart cells, so that they know what normal looks like and they can compare them with disease states. So we're going to jump right into the book. I put this QR code on here so that you can send me pictures of what you see, because I love seeing what you find. If you see something real cool, it's sometimes when you look at cells, it's like looking at clouds and you go, oh look, it's a turtle, it's a squid or something like that. If you have a really good image that you could be taking with your camera, send it to me. Let me know. I do want to let you know that if you send me something and I think it's cool, I will probably put your name on it and then show it to other people too. So just know that. Let's go over here to our book, page 1. So, Microscopy 103, we're looking at the cells themselves, and let's go to these notes. Oops. I messed up my notes page. There you go. So when we study cells, there are really three things about cells that make them alive, and if they don't have these three things, they're not really alive. So the first thing you'll notice about cells are that cells everything that's alive consists of cells. Everything that's alive consists of cells. So living things need cells. You have to have cells. If you're smaller than a cell, you're not alive, which is number 2. Cells are smallest units of life. Nothing smaller than a cell can live. Cells have to carry out specific functions. They have to be able to eat, they have to be able to breathe, they have to be able to replicate, they have to be able to move around. Sometimes it's tricky with trees, you think there's in one place, but we'll talk about that when we get to plants. But these are the smallest units of life. They're able to sustain all of the properties of life on their own. So one individual cell in me, a neuron in my brain, eats, it breathes, it can replicate at some point in life. It moved at some point in its life. It does all the basic functions of life. The third thing is that cells have to come from other cells. Cells don't just pop out of nothing. You don't just throw a bunch of pieces in a beaker and then poof, you have a cell. Cells come from other cells. So what's cool about life is that when you think about it, everything that happens at the cell level happens to you. You eat, you breathe, you can replicate, you can move around, you can protect yourself, you can do all these basic functions of life. You came from 2 parents, right? So your parents shared their DNA and they made you. So cells are tricky because 2 parent cells can come together and they can make an offspring that's just like the parent cell. But you can also have one cell that comes along, works hard enough that bloop, it splits in 2, so it replicates everything on the inside and then divides itself, and now it has a parent and a daughter cell is what we call it, so an offspring cell. They're identical, but they came from something. So cells always come from cells. I have a doctor's handwriting. I'm so sorry. So we're going to talk about really 3 basic types of cells. We'll talk about plant cells, animal cells, and bacterial cells. We've talked about the classification or taxonomy before. Originally, life was classified as animals, because it had like legs or muscles or some kind of tissue that can move around and it's mobile. Plants that were sessile, or they were stuck in one place and couldn't move around, and they were typically green, or it was mineral, which was basically other. So you have animal, plants, and minerals. Boop. We found microscopes and said, hey, these things are more complex than we really thought they were originally, because some of these things that move around are actually plants, which is odd, things around the water. Or vice versa, some things that are stuck in place are actually animals or that don't move very fast. But we got to classify them a little bit better based on their small properties. We also discovered bacteria. So where did bacteria go? Was it a plant? Was it an animal? Who knows? But they came up with 5 kingdoms, and you can see them on the little tree over here. And then along came geneticists, and they said, wait, we look at DNA and RNA, and we figured out that things can be split a little bit better. So they came up with a 3-domain system and took those 5 kingdoms and split them into 6. And what they did to make the last kingdom, they looked at the original 5 kingdoms and said, hey, that Monera, bacteria basically, we'll say bacteria, can actually split into 2. We're going to call them Archaebacteria, Archaea. Put the little dash at the end because your prefix is archaea, and then it's just bacteria. And the U bacteria. U means the standard or the normal. So if I Archaebacteria, Eubacteria, I have these new groups, these new kingdoms. Archaea are the extremophiles. Extremes. They love extreme climates. Extremophile tells you the extreme phile is lover. So they're extreme lovers. They love the hot, cold, whatever extreme you can think of. They love the acids environments that would just melt your skin right off, and the basics that would slime your skin right off. They love the extremes. They love being in the deepest part of the ocean where the pressure is so intense that it would crush a lot of animals. They like being in places that the air is really thin, like the top of Mount Everest. So they're the extremists. The rest, everything else, is normal. So we'll just put everything else. Boom. Eubacteria. So like I said, today we're going to look at some of these different groups and classes. Let's go to the next page. Boop. So you remember the discovery of cells. We talked about microscopes being created, and we talked about 2 guys, actually kind of 4, but the 2 guys that really started looking and seeing creatures and organisms and things were von Leeuwenhoek, who was a seamstress and was really good at polishing lenses, and he saw the animalcules, which were actually like bacteria and protists. And then Robert Hooke, who looked at a cork and sliced it off and said, hey, look at these little structures. They look like monastery cells or amongst living quarters. So we'll call these little square things cells. What's interesting is that neither of them really knew what they were seeing at the time because we hadn't described what you could see at the time. So we'll actually replicate one of them. We've looked at von Leeuwenhoek's before with drops of water. So what we're going to do is we're going to shave some cork. So the next time your parents get a champagne bottle or a wine bottle, you can do the same thing. You have a sharpener. Okay. We're going to call this a microtome because what I did is I actually took the screw out. I flipped the blade over so that it shaves things on the outside better. Tighten it back down, and we can do a little shaving on our own. Oops, that's what we want right here. That's better. It took me a couple tries. So I'm going to take this, and you can take a really sharp knife, like a scalpel, and shave really thin pieces off. They have to be very, very, very thin. And even with a really super sharp scalpel, sometimes that's not easy. So, I'll show you how it's okay to be a little bit messy. Take some of these shavings. I flip my blade over, and I'm just going to run it right along the edge. Oops. So I'm sliding, wiggling back and forth, get a little sliver off. And I'm hoping you're doing the same thing with me because you're going to use your own pieces. Okay, I spun some, you can see it's kind of curling off. Man, that one might be just perfect. So thin. Okay, so I just spun it a little bit, put the edge of the cork in, brought it over and spun. Let's go back the other way. I can feel it. You can kind of feel the roughness. It's like when you sharpen a pencil. There we go. Lots of little tiny shavings. I'm just kind of curling it in. I'm trying the different sides because I noticed that the bevel on this sits a little bit lower. This one sits a little higher. So this top part is taking a little bit bigger pieces. Bottom, not so much. There we go. So we have some shavings in there. Still have some stuck. That's really all you need. There's my cork, my sharpener, and my tweezers. I'm going to push some of these shavings together. That one I'm betting is going to be perfect. Alright? So here's what I've observed. I've observed pictures of cork cells, and they're kind of square, if you didn't notice. But I shaved a bunch of curly perspectives, so I'm not always going to get the same slice, right? So here's what I think. That's what I hypothesize. I think the cork cells are not exactly square. I think I might find more shapes, things that aren't square, but maybe they're pentagons or hexagons, or maybe they're triangular, or maybe there are some round ones, but they're not all going to be square. Right? My experiment, I took cork shavings. How can I observe these? What skills have you learned? You could take a slide, you can make a slide, whether it's a quick slide out of some transparency paper, or use a plastic slide, or use a glass slide and cover slip. So let's say cork shavings, I'm going to use a glass slide, and then I'm going to try a dry mount, then a wet mount, and of course I'm going to try out all three objectives: scanning, low magnification, and high magnification. If you want to get crazy, you can add some stains to it. Put a little methylene blue, put a little bit of iodine. Will they work? Well, based on how methylene blue, what it binds to, proteins basically, and iodine that likes like cellulose, carbohydrates, which one would you probably use in this situation? You predict. So that might be your next experiment. Once you find out if they're exactly square or not, repeat it, but we're going to say that methylene blue binds to the proteins, and we'll be able to see what inside. Or we'll try with iodine. Iodine binds to carbohydrates, so it'll bind to what inside, make it stand out. Okay. So our wet mount and dry mount. I'll take my glass slide. Oops, which I just had. There's my cover slip. Don't forget, when you take the cover slips apart, hold them by the edge. Be very careful. I'm actually going to wipe off my cover slip. Go in the same direction. If I see some fingerprints or something, I might clean it with some Windex. And I'm going to come over here to the toolbox. Glass slide. Put it down. I'm going to take my tweezers, and I saw that really thin piece that I really liked, super thin. I mean, I can see paper right through it. I'm thinking that's going to be perfect. But I'm going to take my finger and kind of brush some of these loose pieces together. Press my finger on it and just sprinkle them. My finger's a little dry. Okay. Back to square one. Don't really want to crush them. Let's try something new. I will just push. It's like a dustpan. Push some of these shavings. I don't really want the thick shavings. I just want some of those thin ones because I'm going to put some drops of water on it in a few minutes. So I'm going to check it out. Dry mount first. Alright, I'm going to bring it down, make sure that my red scanning objective, if you don't remember, red scanning, I guess I could bring this over here so you can see a little bit better. Right there. I'm going to bring my stage all the way down, take my tissue paper, this. I'm not hitting the lens, I just had some stuff on my stage because I want to make sure I use lens paper, and this is actually a Kim wipe right here. But I have lens paper too, just haven't used it yet. Everything looks pretty good. Actually, looks like I have a little something on there. So I'll take my tissue paper, lens cleaning paper, sorry. And this is good because this is the first experiment anyway, so we may as well clean that lens. I have something that's sitting on the lens. I'm not really sure what it is. It looks like a speck of dust, so I'm just running across. Yep, I think I'm okay. I'll get my iris on the bottom. On here, I'm just going to drag it across my lenses, my objectives. Can't reach it. Okay. Alright, and now back to the red objective. Stage is in the low position. I have my cork slides. I'm going to put them on the stage. Put my thumbs on here. Stage clips. Bring it on up. Centered in the middle of the diaphragm. Iris. I have my overhead on, which is beautiful about your scope. So I flip the overhead on, and let's see. What it looks like. Down, so I'm going to bring it on up. Oh yeah. Oh no. Autofocus is on. Okay, a problem I've already bumped into. Got a lot of light there. That's better. So what I just did is, you may not have noticed, is that I have a bright light. You can see how bright it is coming from right there by the camera. And it's actually washing out my stage when I flip on my overhead light. So I turned off my lights on my microscope, and this is good enough. You know, you can do this too. If the light on your microscope's not perfect, you can always grab a second light, kind of like this one I bought at the dollar store, and you can add your own lights like I did, coming at whatever perspective you need it to. So you can control your shadows from any perspective. Right, so that's pretty nice. I can see one. I'm going to take a little closer look. So I have a pretty decent focus there. I'm going to spin my revolving nose piece. Right, went from scanning. Now I'm at the 10x, the yellow objective. So I have 10 up here, 10 down there. I'm at 100 times magnification. Pretty cool. Cool, you can actually see already some of the little tiny cell structures. Isn't that awesome? I have autofocus on my phone, which was a bad idea. I'm going to flip this one more, and I probably will lose it because it's hard to use the overhead light, the long objective, and my phone didn't like it already, so let's see what I can do. Here we go. Yeah, it's going to be really hard to do. So I'm going to flip my underlighting on. So I'm going to flip the power switch to the 2. Remember, you have the rheostat over on this side that controls how intense your light is. I'm going to put my hand up here just a little bit to try and block some light. Right, played with the brightness a little for contrast. Now I'm going up and down. That looks pretty decent. But, so this is on dry. I would probably be a little bit more successful if I had some water in there, because remember, when the light goes through the water, it doesn't bend as much as it does through air. Remember the whole topic we did before, pools and fogs, right? The air and fog distorts it so much that it becomes very blurry. The water keeps it pretty precise. It just warps its perspective, so you still get a clear image. It looks twisted. All right, I'm not going to get this on the highest power, so let's go back. I'm going to go back to the surface. Oops, turn that off. Fine focus. Oops, a little too much. Bring it back, bring it back. There we go. And actually, I'm going to move around and find some more pieces, because remember, I had that very thin piece I thought was going to be perfect. Yeah, that's it. That's got to be it. Anything past here? Nope. Oh yeah. Oh no. I lost my camera again. So I'm having a little bit of technical difficulty with my cameras lately. Still see those cell structures. That's really cool. In fact, there are some places that are really thin, so I'm going from the scanning, 40 total magnification. Remember, 4 down here, 10 up here. 4 times 10 is 40. I'm going to go to the low power. Just the fine knobs, remember? Oh, there it was. Awesome. Look at that. I'm actually going to snap a picture of it while I'm in here. Oh yeah. Got it. So it's nice because I can kind of control my autofocus. Those are pretty good. If I do say so myself, and I actually am saying so myself. So, what I'm going to do, I'm going to brush this out of the way, grab my pencil, and I'm looking at yellow objective, that's 10. Doesn't have to be perfect, but remember I kind of curled the cork as I went along, so if you notice I have kind of like a swirly pattern coming up here. see a couple of those hollow areas. So this one's actually kind of like a half moon. I have a few randoms. And I'm not going to draw every one of these. I've got a little gap down here. But what I am seeing are that these cells, cells on the bottom left, as they swoop up, kind of have through here a rectangular shape. So I think it was kind of right. I'm only really drawing that part over here, but then I have a couple of these odd ones here in the middle, and this was at 100 times, and I undrawn. I didn't draw that side. Okay? But it's kind of cool what I saw here. What I'm going to do now, flip to my scanning again. I'm going to bring this out. And then I put this here so that you could see it. But you can always flip to the next page, and the next page talks about wet mount. So with a wet mount, I'm going to take a cup of water. There's my eyedropper. I'm going to put some water in the eyedropper. So I'm going to squeeze the eyedropper, suck the water in. Have a few drops there. I'm just going to hover. Boop. Ooh, man. I probably should have stopped at 1. I'm going to stop at 1. Slide cover slide, sorry. I'm going to take this, I don't know how well you can see it, but here's the edge. I'm going to tilt it at 45 degrees, touch the glass, and I'm just going to drag it back until it hits the water, let it drop. Boom. Wow, that really filled about all of it. I don't think you can see all that well right now, but there's a little bit of air over here on the side. So what I'm going to do is I'm just going to touch the side of this. A little bit of water. Boom. And I didn't have to put a full drop on there. I just touched the side and it sucked the water right in. Right. I'm going to bring this back. Oops. This piece is all under there. I don't want air bubbles, and I just created another air bubble, so I'll just put a little more water here. There it goes, perfect. And if it dripped off the edge, I would just take a paper towel, tissue paper, whatever, and clean the edge off. So I have some cells. My goal is the same. I'm bringing my stage down. Thumbs popping up the stage clips. I'm going to center that really nice one right here. See it. Right. I'm going to turn on my camera again because it likes to shut off on me. Boop. I don't want to see my face up that close. There we are. Okay. I want to see a bigger picture. Start bringing it back. Raising that stage, getting it close to the scanning. Remember the red lined objective. Scanning. Getting closer, getting closer. And hello. There you are. Alrighty. What have I found? Is that my thinnest? We'll look around. Those are nice ones. Remember, I'm still on scanning, but look at that magnification. Crazy. Jump over here. I know I saw some more over on the edge. There it is. There a couple. Ooh. Let me see what I can see here. So I'm still looking from the top. I actually have no lights on my microscope right now. We were seeing that really well. So I'm going to have to block some of this light just so I can get some contrast. Oh, here we go. Lens paper book. And I'm going to turn on my bottom lighting, the number 2 on the power. Okay. Blocking it. Whoops. Sorry. There it goes. Scan this up a little bit better. Too much light. So a couple problems you have when you're working with water, by the way, is you don't have a lot of time because the light is bright and it's hot, and it will cause the water to evaporate, and then you're back to square one. Oh, scope just turned off. Sorry. You just keep looking. I think I'll be able to see some decent cells in here. Okay, I'm going from the scanning objective up 100. Fine-tune knob only, not big coarse one because we don't want to break anything. Sweet, look at that. Oh, too far, Shannon. That's all right. I'm going to slide back over to that big thin one that I saw before. I actually liked the craters without the water because the water kind of fills in all the holes. Ooh, that might be a decent spot. Notice we're going through depth. So I can see the top clearly here. Go down a little bit more. I see the bottom more clearly. I think that might be a really good spot. Oops. Let's go back. We're right here. Fine tune it. I'm going to take a peek from the bottom. I'm going to block that light again. You don't have to do this, remember, I'm doing it because I have a very bright light that I'm looking at over there. Oh, lost my microscope again. Dang it. Alright, I do believe my image was better without the water. Yeah, for sure. Oh, actually that's pretty cool right there. So that bottom left, it's almost like a skeleton. I'm going to try and go one more up. Oop. See that black circle up here? Oop. That air bubble. I'm really more interested right here. I'm going to snap a picture just for funsies. And now I'm going to go up to the highest objective. And I really want to try and see that little, it's almost like an island, you know? Oh, did we get right there at the air bubble again? So we knew the air bubble was up. Let's see. Oh yeah, that's an air bubble right there. Snap a picture, quick. Boop. Air bubble. Air bubble. I'll take it. Right, I'm gonna follow the hook right down around the corner. I don't want to push too far. I'm at a very high magnification, so remember, if I push a little bit too hard, I'm gonna lose it. And I might be losing it anyway. Can't get my finger over there. Dope. Dope, 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 dope. Ah. Oh no. I think it's gone. I ruined it. Oh, look at that. There it is. That's pretty sweet. Slide it over. Come on this way, come on this way. Oh. Every time I go to the stage, it moves back just a little bit. Move it up a little. Too far. Down a little. So if you really enjoy the microscope and you want to get a different one, my suggestion is you get one that has stage knobs. That's really like an Etch-a-Sketch. Because you turn them, they'll move the stage forward and backwards for you precisely, without worrying about your thumbs messing everything else up. Like me and my fat thumbs. Oh no, did I lose it all? Okay. Up, 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 wrong way. There. Last try. Last call to see it all. Other way, Shannon. There we go. Okay, that's pretty cool. So I can see a couple intact cells. Here's a decent cell. There's a decent cell. There's a decent cell. So it looks like they were probably at one time squares, but no, not so perfect. Snapping a picture. Nice. Alright, so what do we find out? And I snap pictures because I want to keep moving on with this. Not all look square. So I could go back and I could experiment with a drop of iodine, drop of methylene blue. What I'm going to do right now is I'm going to flip, just for time, you can pause and do this, but I'm going to flip that blade back over so that it's a regular pencil sharpener again. Notice I changed my color too. And I'm actually going to shave a little piece of the pencil. Doing it nice and lightly, not cranking in. Take a couple more thin shavings. I know you did this when you were little, and you got your hands on that pencil sharpener. You tried to see how long you can make the curl. Yeah? I'm not that talented anymore, but I think I held my grade school record for the longest curl of a pencil sharpener shaving. That's not true. Okay. So, you see the thin shavings? Super thin. You can see right through them. Whoops. See right through them. So I would repeat the same thing, except this time I'm going to say, I can see a pencil shaving. I can see the pencil's tree cells. I'm going to try it dry. Get my scanning up there. Boop. Open it up. Okay. Get my scope on. I know I'm already looking at pencil. That's kind of cool. bit different than cork. This almost looks fibery, like stringy fibery. Ooh, there's a splinter waiting to happen right there. You see it? I'm gonna lift the stage clip up. In fact, I'm gonna take my stage clips and turn them out to the sides, because I think my slide will be okay. Oh, there's the tip. Cool. You can see some graphite on there. That's decent. Turned off my overhead light. Now I'm just using the room light. I like that. This was just at the scope. Pretty sweet. I'm going to turn my revolving nose piece. I'm going from the scanning 40 total magnification up to 100. Fine knob only. Fine focus knob only. Thank you for not breaking your own stuff. Nice. Went a little fast. Very cool. Layers, like when we looked at the newspaper. So this all comes down to depth. There you can see the parts that are closest to me are clearest. I'm going to go a little bit deeper. Now the ones that are a little further away. So I'm looking here, peak of the mountain, mid-mountain, heading down towards base camp. And I lost my image, of course. The timing, incredible. Boom. Don't want to see my face. There's my hand. There's my shavings. There's base camp. Alright, I'm going to do one more. Actually, let's see what happens when I turn on underlighting. Can I see some cell structure? No, not really. So what do you think? Do you think this was a real piece of wood, or do you think that it was compressed from something? I mean, based on what I was just saying, I would guess compressed, but we did see some stringy fibers. Yeah, it looks pretty shredded and torn, really, right? It doesn't look very like cells. I'm going to go crazy. I'm going to go all the way up to the highest magnification. Oh, yep. So crazy, I actually just pushed my shaving right off. Experiment, see what you can see. Maybe break this up into smaller parts, put it back under. With a drop of water, see what happens. Add a little bit of iodine. Add a little bit of, yeah, very fibery. Add a little bit of iodine, add a little bit of methylene blue. Yeah, it's just like splinters. I think this might fall into the textile category, where you have small fibers of cellulose compressed into a textile. Perfect timing. Okay. So that was our looking at wood and cork cells. I'm going to brush these. Oops. I don't know where I put my garbage. So I'm just going to brush them off into this napkin, because I really don't want to have pieces of it all over the place later. There we go. I have my little trash pickup. And we can flip to the next slides. Oops. Is that what we want? We want that one. So we did the temporary wet mount. I walked you through it. You can see the different steps. Keep in mind, if you're working with a live creature, organism, that it's going to be hot for them. It's like putting the sun right on you. And you're not going to like it, so they're going to try and get out of the way. The water's going to evaporate at the same time, so you really have a short amount of time to work with them. You can add a little bit more water from their source. Don't add distilled water to them. Add the same kind of water that they were with to it. And then with staining, we talked about it before, you can put a drop of stain right on the side of the slide with the water, and it pulls the stain right in. I'm going to pull this off. I'm actually going to I'll put it here. But I'm going to take a piece of tape and I'm going to tape my shaving down to my page so that my notes stay with it. Alright, let's go to the next page. Cell replication. So with cell replication, you're going to look at a prepared slide of an onion. If you open up your box, cells, this one, remember you already wrote top here? Smart. Open it up. Find number 32. Number 32 I can see down below. Slide the edge up. Wow, look at me go. 32. Onion epidermis, whole mount. Take my tissue paper, brush it off one side, brush it off the other side. So, cell replication. When cells replicate, I mentioned this earlier, so let's look at the whole thing. Boop. You can see in the top that cells can start from one cell, the oval, and then bloop, turn it into two. By binary fission, pulls apart, and you have two perfect replicas of whatever this was. I watched the movie Evolution with David Duchovny the other day, and I love it. There was actually an episode of Big Bang Theory, same idea, where Sheldon starts shaking it and bloop, he turns into two Sheldons. But that's the same idea, is that you have two copies of exactly the same organism. Everything's the same. The DNA and everything are exactly the same. So a problem with that is that if this organism's in a new environment and it replicates, but the environment's hostile, its offspring is exactly like it was, so it probably doesn't have a really good chance of changing or evolving for that environment to grow and live. So two forms that we use, humans, animals, plants, are meiosis and mitosis. So meiosis, I always see the I in the middle, and what it does is it'll take a cell and it will make individual baby cells, so that every baby cell is individual. It has unique characteristics, and this is a huge process. This is like when mom and dad give you sets of genes and you scramble them up and you take certain pieces from mom and dad. Super complex, and we'll probably have a video on that some other time. So one we're looking at right now is mitosis, and I see the T in the middle of it, T for twins. So the parent cell will do similar to binary fission, where it will replicate all its DNA and all of the pieces inside, and then it will put up a wall, so you have this round cell, pinch down the middle, and then boop, you have twins. Twinsies. And that's mitosis. And there are different stages. Interphase, if you look at it, remember the parts of the cell, the center part, you have the nucleus with all the DNA inside of it. Interphase, this nucleus becomes very dense. So normally it's really thin because your DNA is spread out all over the nucleus, so the light's evenly split apart. But now the DNA is coming together almost like fingers. So before it was all cloudy, now you can see the individual fingers coming apart, and that's interphase. Right. Next, prophase, you're going to see the nuclear membrane, the wall, shouldn't say wall, the liner around the nucleus starts breaking apart because that DNA needs to spread apart and go into these two separate cells. Okay. Next, we have metaphase, and metaphase has a special characteristic called a plate. So with metaphase, you're going to see this DNA that's coming out, moving into, oops, sorry, this might be a little better. I think you can see my mouse here. So you can see this DNA is starting to line up right across the equator or the middle of the cell, like that. Right? That's metaphase. Next, it's getting ready to split apart, so you're going to see this DNA that's all lined up actually rip itself in half. So half of the DNA is going to one side, the other half is going to the other side. You can say north and south, east and west, top and bottom, left and right, whatever term. It's a round cell. There's no real up and down, right? So the DNA is going over here, making a new nucleus. DNA coming over here, making another new nucleus. This is called telophase. And the last thing is cytokinesis. So it's where the cell will pull and pull and pull. You get that parent cell and whoop, pulls apart. Cytokinesis. So if you look at an onion, while it's starting to replicate and grow new cells, you can see these different phases. Onions are nice because they have larger cells. They have decent sized nucleus. And when it's prepared to slide, they've already stained it for you, so they've done a lot of the work. Let me turn on my microscope. Camera. Oop, flip it over. Right, I have my stage in the low position. I'm going to bring my stage clips back. Right here, stage clips. I'm going to slide down into the middle. Okay. And I'm going to look at, my scope again. Wow, look at that. Green, so green. So you can stain things real green too. Pet stores have it for aquariums. It's super green. Okay. I'm going to turn on the light below. Swoop. Look at that. So cool. Uh-oh. I'm not seeing a lot of nuclei. I got a bad slide. I'm going to go in a little bit closer. So I'm taking my revolving nose piece and going from scanning to the low power. Using the fine adjustment knob, bringing it in, bringing it in. Oh no. That's no good. So my root tip is not so tippy. But you can see all the cells, right? So I'm going to do something a little bit different. I actually sent with you some minced dried onion. So what you can do is get one of your clear plastic bags. Just like this. Take some of that minced dried onion, put it down in the plastic bag, fill up with water and leave it. If it's warm water, probably like a half hour on the table. And before you know it, you will have dried onion. Poof. Not magic like you see in the movies, but it'll work. So I'm going to take a slide from earlier. Here. I'm going to take my tweezers. Here's my little bag of onions. Really smells oniony too. I'm just going to push them on against the wall of the bag and drag up. couple little pieces of onion. Little tiny slivers. The thinner they are, the better. Okay. I'm going to take a drop of water. Slide. Take my cover slip. (45°), drag it over. Drop. Okay. So it looks like one of my pieces of onion is too thick. I'll take my probe. Brush. Bring it in. I'm going to get that one big piece out. See ya. Boom. Okay. So now I have just a couple small pieces. And I'm going to take a peek at this. I'm going to put a little bit more water in there. So I see a bubble right along here. I don't know if you can see the bubble. Water right here on the edge. Oh, still a little bit of bubble. Bring my other slide out. Going down to my scanning again. Lift up the stage clips, pull it in, turn on my scope. Right away, there's some onion. Oh, maybe not. Nope. That's a little more oniony. Sweet, look at all those cells. That's underlighting. There's overlighting. Still pretty cool. Nice. I'm gonna go along. Oh wow. That one. That's pretty thin. That's pretty decent. Period. Very cool. I'm going to snap a picture of that one. Like that, you can see some of these individual cells here. Boop. Hello, photo op. Right there. That looks pretty good. This is still on scanning. So I'm going to turn my revolving nose piece over to the 10x, fine-tune knob only. There's no white. You can see a couple bubbles there. See the bubbles. Turn that light back on. Sweet. Seeing a lot of cells with no nuclei though. I can see some fragments of things in between. So it looks like we have a lot of pieces of onion that are not at the root. All right, what I'm gonna do, I'm gonna grab a toothpick. Oh, maybe. So today we're learning what happens when we run out of supplies. You improvise. So I have my pick here. Found the pick. Methylene blue. I'm actually going to go back to my scanning for this because I really want to watch what happens. I love watching diffusion. I love it when a plan comes together. I love it when diffusion happens. There we go. Right. Here we have just plain lighting. Right. You don't need a lot of methylene blue. I have a big bottle here. So I'm just going to take a little drop. Ooh. Add it to the water. But not quite enough. Oh, and I lost my camera at the same time. It's not as good as the toothpick because the toothpick's slightly, there it goes. Gotta get that camera back on. Just gonna keep adding a little bit at a time. I can see the blue creeping. Put that back somewhere safe. Whew, I didn't get any methylene blue on my fingers. That's awesome. I like that. Right. So I have, you can't really see it I guess from this perspective either, but I've got a little puddle of methylene blue sitting over, but there's a bubble or something that's isolating it. So I'm going to take my dropper again. Oh yeah, I do have a little. I'm just going to put it right next to the methylene blue, and that water just pushes the methylene blue right in. Just kind of flushed it. It's creeping along, slowly but surely. I'm going to go over and look a little bit. I can wait. I'm just going to look right at those cells. Here we are. Let me snap a before picture. Right. Oop. Right there. Cool. Where can we get those? It's pretty decent. I'm not seeing what I want yet. So I'm going to go to a higher objective. While methylene blue creeps along. I can always put some more in and it'll go faster. Fine tune. Uh-oh. Oh no. Did I lose it? Okay, I'm going to go back to the scanning. Let me find it again. There it is. I like what I see. I'm going to go to the yellow 10x, so 100 times magnification total. Easing around, easing around. I think my autofocus on my phone might be losing it for me. So one more shot. I go back to scanning. Find it. Wrong way. There it is. I'm going to move it down. So that's really what my camera is looking at right here. Boop. Right there. Flip it one more time. Alright, remember, when you push on it, push the opposite direction unless you're going up and down. There we are. Come to focus. So cool. So I can see the shredded cells. This was minced onion. So there's a shredder destroying it. No nucleus, no nothing. All I see is the outer wrapper. All through here. Wrappers, wrappers, wrappers. Okay, gone. So, one more thing I'm going to do. I'm going to take my pencil, just like this with the eraser. Turn this back to scanning. Need to focus it, but I'm going to go right on top of that piece of onion. It's been soaking a little while, and I'm going to kind of smush it a little bit. It's not smushing too easily, so I'll pull it out and get a nice clean smush. You can see my dyes, flat down on it, smush, smush. These guys are holding it up. Smush. So I'm going to slide this down, give another smush. I don't want to get mess. Blot up the excess here. Put it back under my scanning. What do I see? Nope. Over. Let's see. Underlighting. Overlighting. Way overlit. Right there. Move around. Cool. A little bit stronger power. Turn the revolving nose piece. There it was. There they were. Oh, just when you think you got it, you lose it. Click and click. Oh. Want to go in and look a little bit closer. Oh, look at a couple of those cells with nuclei. Can you see them? There. Photo op. Click, say cheese. Nice. I'm going to get really brave here. Got my underlighting. And I'm going to go one more objective. Click. See how fast it came in, focus on left. I think I see a nucleus right there on the left, so I'm going to push it left. Nope, that's a bubble. Oh, what's that guy? Right there. Underlighting on. Adjust my rheostat, see if I can adjust how much light there is. My autofocus on my camera is still messing up. So it looks like I'm not going to get a good picture with my autofocus on. Oh well. So here are a couple things you can do. You can take an onion from your kitchen, just thinly peel the surface off. So take that brownish color layer if you have a white onion, peel the outer layer off, and then just take your tweezers and peel a real thin piece, not the whole chunk. So when you're cutting an onion, you can pull a whole layer off. You don't want the whole layer. You want the surface of just one of those layers and slide it under and do the same thing you just did. See what you see. It's kind of fun. Right? So let's go on to the next one. Right. On the next page, we put a list of stains. On the last page, you might want to put a note on, not all the experiments go the way they're planned. For instance, this one we couldn't even see the nuclei. We got too high up, and we'll talk about plants later, but we got too high up on the plant, is where it looks like they sliced, instead of at the root, where the cells are rapidly multiplying to grow. Right? The older plants, just like we saw with the cork, they had no nuclei, because the outside part of the tree, the cork, and if you haven't watched it, watch, how do they make cork from a tree? Corks from a tree. And you'll see that they just punch these corks out. It's actually a slice of the tree, but it's a dead outer layer that's right underneath the bark. Same with the onions. We must have been seeing the dead outer layer that's up towards the top. Right? So stains. Here's a list of stains. My piece of advice is that you make your own list. So over on the other side of your notepad, you might write methylene blue. So methylene blue on one side, and then what's it good at staining? Proteins. So anytime we talk about a cell that has proteins in it, this would be a good stain to go with. Iodine, really good for carbohydrates. There are more stains than are listed here, but if you look at the key pieces of stains, you'll find out what's good to stain. So on the bottom of that page, you can see the purples and the pinks. This is a gram stain, so they use a purple dye to grab a hold of certain types of cells, and then a pink dye to grab a hold of other parts of cells. When we get to bacteria, we'll actually talk about that even more. Okay. Sometimes you can use a color filter. On the bottom here, on your diaphragm, down here, you can turn this. When you turn it, the best way to do it is just push down a little bit on it and turn, click. Push down a little bit, turn, click. But sometimes you'll find the right filter to make it pop. Okay. This did not make it pop. I'm pushing down on the rim, giving it a little spin. Boop. Nope, still no pop. Might get unlucky with these. So the cells were already hard to see. Yep. We're out of luck on these. And I'm out of focus. Sounds like a country song, doesn't it? We're out of luck and I'm out of focus. Oh, there it was. Let's see if I can get kind of focused and then click on at you. I see it hiding. Let's change our rheostat, the brightness. That helps a little bit. But yeah, that autofocus is still on and causing problems. Adjust the rheostat. Do my find. Oh, I lost it. Yeah. Oh well. So it looks like I am on the blue, which made it pop. So I have a blue filter, iodine blue. Purple's not too bad. Green, not good at all. That's not so good either. Well, I'm not gonna find it. Okay, so make a list of your own stains. Animal cells. Let's actually take a piece of you, an animal, and make a cell out of you. So if we flip to the next page, animal cells. So for this one, it's animal cells. Animal cells have particular parts of them that you see in every animal cell. It doesn't matter what it is. One would be this purpley structure in the middle. Get a little hand there. So looking at this picture, part right there, the nucleus, that's where the DNA is stored. DNA is like the blueprint for everything that's inside of your body. If you need to make a new protein, boom, the recipe's right there in the DNA. If you need to make new skin, then boom, the recipe's right there in the DNA. It's like a library. I always think of the cell like a little city. It has the library where all the history and the records are kept, all the important blueprints for everything, the diagrams for the buildings, they're all there in that nucleus. Outside, you have the endoplasmic reticulum, big fancy name for blurbly stuff that's inside the cell. So all of this fluid out here, this yellowy stuff, is called plasma. It holds everything inside the cell. So the endoplasmic means within the plasma, or in this yellow fluid. Reticulum is just describing the wavy shape. So you have rough endoplasmic reticulum that makes proteins. So here you have the library, and right outside you have a factory that makes proteins. One of the bulkiest, well, all of the nutrients are important, but a very important structural thing is protein. I think of eggs. Proteins in eggs are runny, but when you change them a little bit, they're firm, like in a hard-boiled egg. Your body's the same way. You build proteins that give shape to your body. Okay? Out here I have smooth endoplasmic reticulum. This is where I make fats and cholesterols and things that are not proteins, sugars. And then over here I have something called the Golgi, and this is my UPS. It's my shipping department for my city. So the recipes and blueprints are here. The factories are out here. The ribosomes are little things in here. They're the workers in the factory. And then the Golgi is the UPS that will take the product from the factory and deliver it out of the cell to other parts of the cell or to other things inside the cell. So the shipping department, some of the places you might ship it might be these mitochondria, which are the energy conductor or powerhouses. So I think like a nuclear power plant here that provides energy for everything else in the cell to run. Okay? You might have these little peroxisomes that are moving things back and forth. Peroxisomes because they have hydrogen peroxide that breaks things apart. So they're kind of like the cleaning crew, the garbage disposals. So they destroy everything that's unnecessary in the cell. And then of course you have the membrane around the outside that your cell is actually a dome. It's a city inside of a dome. Right? So those are some of the basic structures in an animal cell. Oops. Here we are. Oh, now I can't move it up and down. Ah, there we are. Great. So all the cells, this is a generic blob, all the cells have these parts, but they might be for different uses. So you might have muscle cells that are all the same things, but then you have a lot of mitochondria for energy to contract the muscle. You might have a neuron like this that has lots of proteins that it uses as signal senders in here, giant nucleus. And then you might have a weird cell like this, the red blood cell, that actually doesn't have a nucleus anymore. It's a long story, but we'll save that for the blood. And then if you look at other cells like epithelial cells, the skin, their special shapes create a protective barrier. Adipocytes, there's this nucleus right there. This is a big blob of fat. It's like a storage unit. So it still has all the organelles in it, all the little structures. It just has its own shape for purpose, the same way a neuron does, fat cells do, skin cells do. So we're going to look at some skin cells. We're actually going to peel some skin cells off of you. So the epithelial cells, epithelium means a sheet-like covering. There you go. So sheet-like covering, you can see over here, you have the flat little tiny layer. Here you have the flat multiple layers. So we name these cells because if they're flat in one layer, they're simple. If they're multiple layers, we call them compound or complex. Right? So if I have something like this and I look at the shape, they're little flattened cells, so they're squashed. I would call it a simple squashed or squamous cell. Here I have multiple layers, they're all squashed. Actually, the surface layer is squashed, so I would call that compound, squamous. All right, we're going to go to the next page and pull some right out of you. So you can see the blue cells there are actually human cells. What you're going to do, you're going to grab a slide. I have a clean slide. I just touched it with my fingers, so I'm going to have to wipe it off now. Got it. Right. Get a clean toothpick. I just got a toothpick out of a box. I've been holding it in my finger here. Put your slide down. You're going, oops, let me flip the page. You're going to, great, not poke, not jab, these can be sharp. You're going to scrape, push to the side of your cheek and drag down. A couple times. Get a little bit more there. I kind of smeared it. You can't see that very well. This, just make it a little bit more visible. Do it a few times. I got some more there. Just kind of smearing it. This is actually called a smear. Just rubbing it back and forth. You can see. That toothpick is trash. You put one drop of water on top. Hovering, I don't want to touch my, there we go. I don't want to touch my eyedropper to the glass, so I hover. And I need my cover slip. Where did I put it? Get a new one. Grabbing it by the side. Got it. Okay. Bring it down to 45 degrees. I know it's hard to see. Dragging it over until I hit that water puddle. Dropping it. Ooh, perfect. All the bubbles are out. Doesn't look like there's any excess. Okay. I'm going to experiment. up to my red scanning objective. Put that aside. I'm going to put this slide on the stage without the pink. I just used the pink to give it some contrast. Ooh, there's that word again. Okay. Turn on my bottom lighting. Oops, there it goes. Turn it up pretty bright too. Make sure that I have camera on. Slide it in. Ooh, man. Got a lot of something right there. Nailed it right away. Cheek cells. Do they look just like the picture? Nope, not at all. Because those cheek cells in the picture are probably at least 400 times magnified. So right now we're on the scanning, so we've got 4 times 10 at the ocular. So it's 40 times. I'm turning my revolving nose piece to the yellow, which is 10. So now I'm at 10 in the objective, 10 in the ocular, 100 times magnified. You see the shapes? Especially right up here. See the cell? See the nucleus? Nucleus down here. Yep. Pretty cool. Alright, I'm going to be brave. I'm going to flip the revolving nose piece one more time. Oh yeah. Look at that. You see the cell. I'm going to look around and see if I can find some others. Air bubble. Oh, is that a decent cell? What do we have here? Oh, I missed. I lost it. Oh, did you see that? What have I been eating? It's like Jaws down in there. So you'll see some movement of things because of the water. But you might actually see organisms. Your mouth is filthy. This looks like I'm touching the glass a little bit, so if I move, I start losing it. There's a decent cell right there. You can see its nucleus. You can see its cell membrane going around. That is not a living cell. I'm touching the glass. So there you can see its nucleus, cell membrane. Looking for little tiny guys like bacteria. Oops. All right. Alright, so I'm looking through the 40x objective. I've got my 10x ocular, so I'm at 400 times magnification. I'll look around. I'm actually going to come back to my 100 time magnification. There we go. Oh, look at that. A couple of decent cells. There's a pretty nice looking cell right there. Nice looking cell right there. See it? Change some of my filters. That was on blue. I accidentally left it and forgot. So greens, yellow, purple. Let me check. And here's no filter. Okay, now I'm going to take methylene blue and do the same thing we did before. Come off of this. So I have some toothpicks. I'm going to grab a nice clean toothpick. Open my methylene blue. Always make sure I know where that's at. Very messy. A little drop there at the tip. I'm going to reach down and touch the water. Dry. I'm just going to add a little bit of water with methylene blue here. I put the methylene blue down on the slide, hover my drop of water, let it roll right down the methylene blue toothpick. There we go. I can see it diffusing already. Out, take a little peek at it. Center a little bit. Tap it down. Put it back under. Scanning. There's my edge of my slide. Oop, lost my microscope. Boop. Boop. There's my face again. Don't want to see that. I see it every day. Get sick of it. Let's see if we can figure out what that is right there. Might be just a little of my lunch. Oop. Bring it in. Nope. Just some debris, a little lunch. Those cells. nice cells. And I lost him. There's a cell. Another cell. So my methylene blue needs a little bit more. I'm going to pull this back out. I know where those cells were. Take my toothpick. One more methylene blue. And right in there, tap, tap, tap. See it? Nice slide, 45 degrees, boom, drop it down. One more, and take it another check. Turn on my camera again. Let's see what we find. There's the edge of the slide. Decent looking edge. Okay. Bring it down. Lots of cells. Huh. Air bubble. There are a lot of cells. See them all? Nice. Okay, I'm on scanning. I'm going to go in a little closer. There you go. There are 3 cells. Several cells there. So I'm looking at 100 times magnification. Now what I want to do is figure out, can I get a really good image here? Keep touching my slide. Cool. Oh. There you go. It's kind of a cluster of cells. You can see them. You could see them until that went away. Okay, then. So, if I were drawing this, tapping the glass, and I lost it. I lost it. Oops. Yep. Come back. Air bubble. Clump of cells. That's a big bubble. Huge. I can find some cells without the bubble. Looks so much like they're moving around, but they're not. I mean, not on their own, they're not. Lost them again. Alright, I'll stick with that guy. There you go. Get my circle. And my nucleus. Oh, lost it again. Darn it. So maybe we can adjust the white with the rheostat. A little clarity. That's still decent. Up. A little weird nucleus. And then if I wanted to, you can see that the size of the image in the picture way closer up. So I can grab a different eyepiece. Here I have the 20x. I'm going to go back to scanning. Look at all those cells. So water is moving, the cells aren't moving on their own. And it's going downhill, it looks like. So I'm going to switch my eyepiece, my ocular. Pull that off. Pull out the 10. Down. Pop that 20 in. This gets trickier because the 20 doesn't hang quite as well. So now I have to focus with my eye first. We have some decent ones. So I have 20 up here, 10 down here. So 200 times magnification. Gotta make sure the lights line up. Just like we've done before. Photo. Oh, and I lost it. Bad timing. That's what I was afraid of. So this camera actually has that area that's too big. Camera itself to be able to get in the circle. Not going to be able to line it up. So here's your task. I can see it with my naked eye. Some really nice cells. Oops. So you can flip. Said right now you're 200. Flip it over here. Oops, I'm sorry, that was 80. This one's 200. I focused. Now I'm going to go to the 800. 20 up here, 40 down there. And it gets a lot darker. So I'm going to turn the rheostat over here to the side, up way high. Slowly focus. I can see one cell. You should be doing the same thing with your scope right now. Big old bubble. Always on the fine tune. Nope. Start back at the beginning and go back through, starting at scanning, get it nice and clear, go up to the low power, get it a little more clear, and then go up and see if I can get it at 800. But do it. So I'll pull this back out, and we'll go on to our last project. I'm going to put the 10x back in. We're on scanning, pulling my slide out. We'll do a permanent mount later. Part of my camera there. Methylene blue binds to proteins around the nucleus. And the last one for the cells, we'll look at some plant cells. Okay. So plant cells, pretty much the same as animal cells. You can see that they have more of a rigid shape to them, where the animal cells did not. Oops, one more up. There we go. So animal cells have a plasma membrane. Plasma is fluidy. It's a membrane holding everything in. Has a nucleus, all of the organelles in between. When we look at a plant cell, it has a plasma membrane, but it has a cell wall on the outside. And when I think of walls, I think of a house or a building where you have firm, flat, straight edges. So you can see, it doesn't have to be a square. And it can be a hexagon, but you have these relatively straight, flat edges for the cell wall. Still has a nucleus. It has some special things called chloroplasts, which are green floaty things that carry out photosynthesis. So a plant cell is an autotroph. Auto means self or automatic. Troph means nurturer. So they actually create their own nurturing. They take sunlight and carbon dioxide, push them together, twist it up, and it turns into energy. It stores it as chlorophyll. It stores the sugars, basically. We can eat the sugars and get nutrients from it. Onion cells were a really good example. But what we're going to look at for our last one is actually a cast. So let me get the materials. So I've included a leaf in your set. I have a fern leaf here. You might have a larger leaf. You won't have a smaller one. But with the leaves, when you hold them, you can see there's a bottom side and a top side. It's lighter on that side, a little darker on the top. Because the leaf has a cuticle, waxy membrane across the top, pretty smooth. On the bottom, it also has a cuticle or waxy layer, but it also has little tiny cells in it, holes. You can see over here. Oh, the page is blocked up. But on the bottom you have stomata. Stomata are like the breathing holes for a plant, and they're on the bottom of the leaf. What we're going to do is we're going to take, actually I'm going to use this. So you can do it with a plastic slide, you can do it with transparency paper, or you can do it with glass. We're going to create a cast, a mold of the bottom of this cell. the leaf. Fingernail polish. Get that on really tight last time. We're going to take, put a little bit of fingernail polish. Don't go crazy. And you're going to pick one cell. Don't go crazy with fingernail polish. A nice thin layer. Only about a centimeter wide. So the bottom of this cell is about a centimeter wide. If your cell's bigger, or your leaf is bigger, that's better. And set this down for a minute. There. I'll let it kind of start drying. In the meantime, grab your tape. It has to be clear tape. There's the edge of the tape. Dry on the leaf. We cut a little piece of tape. I'm going to take that leaf, I'm going to put that fingernail polish and the leaf right there on the tape. Right. I'm just going to try not to get too many fingerprints on here. So I'm just pushing it down with the back of a brush. And then I'm going to let it dry for about 10 minutes. I want to make sure that it's completely dry, because if I don't, it won't work at all. So you can get up, use the restroom, get something to drink, and I'll see you in just a few minutes. Okay, so it's been about 10 minutes. Remember, you just super glued, basically, or glued the bottom half of the leaf. Here. You put the tape on. Hopefully that quick drying nail polish is dried. We're going to peel this tape off. Slowly. Oh yeah, there it went. Okay, there's our leaf. Okay, so I put this on a toothpick, just like that. You see the outline of the leaf, right? We're going to put our toothpick under here. Let's turn our camera back on. Oops. You're doing that. Perfect. And closer, I'm going to do just the scope. All right. You can see there's edge of your tape. Lots of lines. Those are actually the peel lines, if you haven't noticed. What we're looking for. Here we go. There we are. So these are the peel lines. When you peel the tape off, you hear that look. It leaves those lines, but there is the leaf. Right in there. Wow. There we go. You can already start seeing the shape of the underside of the leaf. Right, so we're on scanning. We're going to flip it. Boom. We're on 100 times total magnification. Clear it up a little bit. Oh, there they are. Perfect. Let me move that down just a little. Oops, too far. Can you see them? Oh, nobody can see them because my camera keeps going off. See them? So looking over here, here are your stomata. There's the opening right there, the stomata, where the cell, oops, right here. There we go. Oh, it's going to keep doing it. Slide that up a little bit. Right there. So there's the stomata. Here are the guard cells. These guard cells, when they get full of water, they firm up and they shut like this. When there's not enough water, they bend, they kind of wiggle like a not very full balloon. So let's say we have the long water balloons. Not a lot of water. The stomata opens and it starts breathing. A lot of humidity, water, boom, those stay shut. Those are called guard cells. They guard the stomata. Can you see them here? See the stomata opening and the guard cells around? Let's see if we can get a little more detail. Oops. Now I stuck to the tape and lost my spot. Let's see if we can find it again. Alright, we'll go back to scanning. Swoop, coming in, coming in, coming in. Other way. Down here. There it is. Okay. Flipping and rotating nose piece. Higher resolution. Sorry, higher magnification. See those stomata? Like little eyeballs there. Wow, look at that. Perfect. Let's go for one more. Oh, we're getting some of that polarizing effect from the tape. Did you see it? Coming down slowly. Huh. Nope. Back up. All right. Let's move this just a little bit. Paintbrush to go up and down. There we are again. Find me some stomata. Finding it right now. Hello, stomata. I'm just using the tip of the paintbrush to help me move. Scanning objective. just figured out the problem. Is my toothpick is getting in the way. Okay. Oh, we saw the stone.